science enthusiasts. Welcome to Spaces Unleashed. Every week on Twitter, we bring an expert to chat live through the Spaces program. And this is bonus content that goes with the Science Pawdcast. We hope you enjoy the show. Fransika, how are you doing tonight?
Hello, I'm very happy that I can join and I'm doing very good.
Awesome. Yay. Woohoo. We're glad you're here too. Chris is just connecting. All right, Chris, I'm going to see if I can move you up to a co-host. We'll see what happens with that. So Fransika, you are now in Canada, right? You were in Germany. Is that correct? Am I right about before?
Yes. So normally I live in Germany, in the north of Germany, in Bremen, which is rather close to the coast. Mm-hmm. But since mid of January, I'm now in Canada and Montreal until the end of March for some research work I'm doing here with some colleagues.
Sweet. Well, we're happy to have you in Canada. One of the first questions we ask is, what got you into science? What was the thing that sparked your journey into science?
Well, I think it just turned out that I ended up with science. So I was always interested in Earth, in climate, in the environment, in the living beings on the planet. And then I started studying geography without really knowing where I was heading. It was just like, okay, I'm interested in the stuff that are taught there, so I might take a try. And then I stumbled into a master program, which was really scientific and looking a lot into science research regarding climate, both reconstruction of past climate and modeling of future climate. Mm-hmm. And that caught my interest. So I decided to stay and do a PhD in a similar field.
So when you were young, like before going into post-secondary, were you a person that was curious about the world?
Yes, definitely. So I was like always interested. I started to be very interested and very in love with animals very early. And from that, I also started to be interested in other natural aspects like nature. And I really enjoyed being outside and stuff like that.
Okay. So in the first half of your education, you studied some crustaceans in a lake. Could you tell us a little bit about that?
Yes, for sure. So that was my master project. So as I said, my master program, it's called environmental history. So it's a lot looking into the past of the climate to better understand the climate system and how changes occur in the system and what are the results from that. And for one thing one could study is the sediments of the lakes. And in there, one can study the chemicals in living beings that can be found there, and that is what I did. So I worked with, as you said, crustacea. The name of the ones I studied is Ostracodz. So they are very small. You can only see them with a binocular microscope. So it's like if you see them with a naked eye, it would look like a grain of sand or something maybe. So it's like a couple of micrometer, 200 micrometers, something like that, a little bit bigger depending on the species. And I worked with those to actually I did not reconstructed the climate, but I did a little bit the work that has to be done before, because if we want to do the interpretation of what we can find in the sediments, we have to understand how the climate is influencing the living species in the lake. Mm-hmm. So that was the goal of my research project. So I worked on shells of the specimens that were just died in the year where we did the sampling. And there were also some that were still alive when we did the sampling. And then we analyzed the chemistry of the shells.
What do they look like, Fransika? These little tiny crustaceans? Like, what do they look like? Little snail guys or different?
No, they actually, they really look like beans. So from the shape, it's really just looks like a white bean. So it's like they are also like bivalves, so a little bit like mollusks. So they have two valves and in between there's a living being. I actually also worked with a few mollusks as well during that study, but the main focus were the ostracods.
Ostracodz, okay, yeah.
But they have two, like, let's say, like a outside shell that is made of two shells, and in between there's a living being, and the shape is a little bit like a bean.
Like a bean.
Yes.
They sound kind of cute, actually. If you compare anything to a bean, that's cute as a little bean. So.
Yes, and actually, so as I said, I had in the beginning, so we had fresh samples from the lake, and as they were directly from the top of the sediments, there were not only the remaining shells of those that already died, but there were also a few living ones in there. And then under the microscope, you can really see that they are moving around, so they can swim a little bit. They have it looks a bit like a swimming arm or something. They can move a bit, and that's really interesting to see.
Little arm, like they have a little scoopy arm to swim.
Yes, so they can move around and inside the sediments, and then you can see the movement, and then you can find them.
Isn't that so wild that there's organisms all around us that you never think twice, but they're just all over the place, right?
Mm-hmm. Yeah, that's true. And they are so small, and you never, when you don't think about it and you don't study them. So I'd also never heard about those before I started the study. And there are so many of them, and so different kinds, a lot of different species. Yeah.
I'll tell you a truth. I know 99% of nothing until I talk to people like you. So there's so much that we don't know until we talk to people that study certain things in the world, right? Yeah.
That's true.
So from your master's study, now we got into this in the podcast, and you're moving forward with more of it. You're studying these Planktick can you say it again? Foraminifera? How do you say it again? I should have reviewed. Go ahead.
Foraminifera.
Foraminifera. Do you remember me practicing it on the podcast?
Yes, I just.
Foraminifera.
Yeah. You can just, so the informal short name is just forams.
Forams. Okay, that's better.
Forams. That's easier.
Yeah.
And, yeah.
Well, that's like my last name is Zakowski. But my students just call me Zed because it's so much easier to say. So we'll just call them, what's the short form again? Forams?
Forams, yes.
Forams. Okay. So these are cool. And you're studying basically where they live, and they produce something called calcite. And that is then, I'll let you finish it. I don't want to bungle it up, but you study these creatures for a reason. Maybe tell us where they are, right? Where are these things you study?
Yeah, actually they are everywhere in the ocean.
Oh, okay.
So you can find forams in every ocean, not in the really shallow area but as soon as it gets a bit apart from the shelf area, then you can find them everywhere. And as you have announced before, so you said that I work with Planktick Foraminifera. So that means plankton, Planktick, that are the ones that are floating in the water column. So they live in the upper, depending on the region and species, but about 200, 300 meter close to the surface. There are also benthic ones that live at the surface sediment, so at the ocean floor, a little bit like the Ostracodz. And there are also some Ostracods in the ocean, but they are more often found in lakes. But the forams are really in every ocean. But in different species, so different species are found in different regions. And my focus is the Arctic Ocean. And as one can imagine, there the number of species is not that high, as the Arctic is, of course, a special region in terms of living conditions.
It's a little bit inhospitable to the average person.
Yes.
The furthest north I've ever been is Churchill, Manitoba. Then we went on into the tundra on Hudson's Bay, and that pretty much the Arctic, I'll say. I think we're pretty close. And that was frighteningly cold. And there's not a heck of a lot that lives there, let alone in the ocean So What do these forams look like that you study? Like, if you could, are they beans? Are they different?
No,
Are they?
no, they are not beans. But
Yeah.
I have another comparable thing that is also something to eat, because what was the first thing I was told when I started my PhD by another former PhD student in our group. He said, like, they look like popcorn.
Oh, okay. Popcorn.
And yes, and really, when you look at them, you can see that. So they are, as we said, they have a calcite shell. So also they have this white, and they form these different bubbles. So they have like bubbles that they add up when they grow. There's another bubble, and then they stick another bubble to the bubble, and then another one, and that make, in the end, they have this bubbly shape with, like, a little bit looks like popcorn.
Okay. Well, you're making me hungry. Beans and popcorn. I don't know if we should. I don't know if those two foods mix together very well, but
probably not the best thing to.
When you say the word planktick, is that related to plankton, like, or different?
Yes. That means that they are part of the plankton.
Really? So, like plankton from SpongeBob plankton, like zooplankton.
Yes. So there's zooplankton, then there is phytoplankton, which is like algae. So phyto, the plants.
Yeah.
And then there are forams, which are a bit in between, because they are not really. They are neither animals nor plants.
Oh, weird! They can't make their mind up.
No, they are just like they are so old, so they are single cell specimens, single cell organisms.
Mm-hmm.
So very, very basic, let's say. And there are some that eat other tiny organisms, and then there are some that have some symbiosis together with some plant cells and do photosynthesis. So they are a little bit a mixture, and they are not really called animal or plant, because they are protists.
Oh, they're a plumanimal.
Yes.
That's what I teach that protists are. Where the biologists were like, "What is it?" And the biologists are like, "We're gonna make up a whole new thing of life because we don't know. Let's call them protists."
Yes.
So how do they survive in the Arctic? Like, it's freezing cold, and like, how?
Well, I mean, in the water, it's not that freezing cold. So they underneath the ice. They live. I mean, still it's cold in the water. But it's definitely much warmer than it would be under on top of the water.
Oh,
in
right.
The air.
Gotcha. Yeah. Okay.
So that is buffered, and as I said, there are different species, and there's this one species that is very happily, apparently, with the colder temperatures, and then there are other species that are only present in the regions where it gets a little bit warmer, or when there's some Atlantic water flowing into the Arctic, bringing some more higher temperatures. Yeah.
Okay. So I'm getting a picture of these things that you study. Now, that's where they are, what they look like. The big question, Fransika, is why are they important? Why are you studying them? What are you hoping to learn from them, right?
Yeah.
Could you tell us a bit about that?
Yes, for sure. So in general, maybe I will start with that. So in fact, even though it's like biological life, it's something that is mostly studied by geologists, and not really, or have in the past not been looked at much by biologists.
Oh. Because
they are so interesting, because they have been around since such a long time. So they have been, as they are so basic, as I said, they are single-celled organisms. They have been on the planet since like forever. They have already been here when the dinosaurs were here and things like that.
What, really? They're that old?
Yes.
Holy cow.
They are like millions of years they've been here. Of course, there has been some changes in species, but in general, forams have been here for ages. like ages in terms of geologist ages, not like human ages.
Yeah.
And that means that so when they build their shell, they incorporate information on the environment in the shell, like the amount of oxygen in the water, things like that, or the chemical composition of oxygen, let's say. And then they die, they sink down to the ocean floor, and then they are stored in the sediments. And that's what makes them so interested to geologists, because we can take out the sediments of the ocean and then dig out the forams out of the sediments, and then do some measurements and really understand what has been happening in the ocean in the past, like thousands of years ago.
Oh my goodness.
So that is really what's make them so interested, and why it has been studied by geologists for such a long time. So that is the general thing that makes them interested. Actually, I'm not that much focusing on, or I'm not really interested in what has happening in the past, but I'm more focusing on what is their role in our climate system, which seems to be a little bit strange, because, as I said, they are so tiny. So they have, like, the Arctic species have a maximum diameter of like 200 micrometers, 300 micrometer. They have a weight of like 2 micrograms, one shell. So they are very, very tiny and very small.
Yeah.
But they are a lot, and they incorporate calcium carbonate. And this formation of calcium carbonate plays a role for the carbon cycle, as well as when they die and sink downwards. So they play a role in the carbon cycle. And we want to better understand what is this role in the Arctic. So how much calcite do they produce? How much mass do they bring down from the surface ocean to the deep ocean? Because there's, like, the ocean has 2 layers, let's say. So there's the surface ocean water, which is in direct contact to the atmosphere, like humans emit CO2, and then it's in the atmosphere, and then it's exchanged with the water.
Yeah.
And then there's the deep ocean. And those two, let's say, those 2 oceans are on short term not in a strong exchange, because there's a like a line in between, which makes the mixing a bit difficult. But like the shells are so heavy, let's say, they are not, as I said, they only have a weight of a couple of micrograms, but still they are so heavy that they sink down relatively fast. And so they can bring the carbon they incorporate in the shell down to the ocean floor.
Oh.
And then so there the carbon is stored in the sediments. Of course, that is playing a role for the carbon cycle in the ocean.
Gotcha.
And so that is what I'm really interested. Like, I want to know how many of them, and what weight of calcite is produced, how many are there, what are the influencing factors on that, and how much is then stored in the ocean sediments, and how that could also probably be changed when the situation in the ocean is changing.
Amazing. Okay, I've got a good picture now. If you're just joining us, this is Spaces Unleashed. We have a guest every Tuesday starting at 7 p.m. Mountain Time. Our guest this week is Fransika Tell. After a brief interview, we open up the floor to questions to our expert, and Fransika has just been talking to us about, I'll say the full name wrong, forams, which are tiny tiny planktic organisms, and you study the ones in the Arctic. So just to recap, just so I get things right in my head, these little foram things, they produce calcite, and calcite is I believe it's a mineral that is formed from carbonates, like calcium carbonate. Am I on the right track? It's like
yes.
What is it? Oh, I'm trying to remember my geology. Is it polymorph or something? Poly something or other, something. Anyways. But the idea is that these little, I might have got that wrong. These little guys or girls, or actually they may not have a sex. These things suck in some CO2 because they're at the surface, and they bring that down to the ocean floor. Am I on the right track? And then thus is the carbon cycle for them?
Yes. Yeah. Well,
maybe I'm wrong. I don't know.
No. You're not really wrong. It's just, it's not that easy, because it's a little bit more complex, because in fact, like, So what they do is they form calcium carbonate, which is CrCO3 as a chemical.
Yeah, CaCO3. Yeah.
Yes, and when during the formation of this calcium carbonate, actually CO2 is released.
Oh.
So on the short term, they actually do not take up calcium CO2, but they release it to the water. But then on the same time, they still have carbon incorporated in the shell, and also then adds up onto this is that they have this calcium carbonate, which is inorganic material, and then inside, of course, there's a living being, which is organic living. And so when they die, even though when they form their shell, they emit some CO2, they in the end also bring some carbon down to the ocean.
Okay. All right. Would you say they're kind of carbon neutral then? Like, do they give off as much CO2 as they suck in?
I think in the end, it's a matter of time scales. So one can say, like, in short-term time scales, they somehow emit more CO2, but on a long-term time scale, they are one of the important features that contribute to the drawdown of carbon to the ocean floor.
This is so fascinating and so complicated. Like I got that wrong, obviously, but I'm trying to think. I get it now. Thank you for correcting my bad wrap up there.
No worries. It's also like I'm always I'm doing this now since end of 2019, and still whenever I think about it, I have to really think about it in a calm moment to really understand, like, how can they emit it and then in the end also draw something down to the ocean. And so it's like the whole carbon system is so complex, and yeah, it's a lot of aspects one has to think. And then in the end, it's really timescales that matter as well.
Right. You have to look at, instead of 10 years, 10,000 or something like that. You have to zoom out.
Yes.
So so they sink down to the bottom of the ocean, and how do you scoop them up? Like what happens? Do you dredge it? How does that get from there to you to analyze it?
So I've worked with different types of samples. So depending on what I want to work on, so I've my PhD project is divided into 3 projects. And in the first step, I really looked at where they are living when they are still alive. So then I look at at samples from the upper 200, 300, 400 water meter in the water column. And those samples I get from so-called plankton nets. So this is like fisher nets, but of course with a much smaller mesh.
Oh, yeah.
Because they have mesh sizes of like 36, 50 micrometers, something like that. And we have some nets that are towed on top of each, or that is automatically, you have a net with like 5 nets on it, and we bring it down to 500 meter and then open the first net and slowly pull it up. And after 100 meters, we close the first net, and then we open the next one and to pull it up again for 100 meter and then close the next one. So we always get an impression of what we can find in depths of, for example, 100 meter. Of course, one can change that depending on where one is studying, what one wants to find. But then we have this collection of the net, and of course we have then some sieving and sorting, like get rid of all the bigger things that we are not interested in, or give it to other people that are probably interested in.
You get some things in your nets you don't want. You're like, get out of here. You're not a forum.
Yeah, probably this is happening. And then so we receive it, and then I'm telling all of it, but actually I've never seen that myself, unfortunately. So because I've never been on a research ship myself this far, due to sadly well-known virus that is out there. But I know the theory, and I know what the samples I finally get. So then those samples are either frozen or they are dried, and then I get the samples, and there I can I put them under the microscope, under the binocular, and then I can just pick them out, take a brush or some other work material, and then just in between some other grains, find everything that I'm interested in.
Grab them with tweezers. It's like a scientific game of Operation. Have you ever played Operation? You got to get the little bones out of the person. I
know the game.
Okay, I don't know if that's a great analogy, but
similar but much smaller.
And if you touch the sides, you don't get buzzed, right? Buzzed, so that doesn't happen.
Yeah, no, but it can happen when it's too dry. So normally I work with a very small brush and some water on it. But when the brush is too dry again, then you touch them and then they just jump away. So because of friction and electric.
Oh, they get static.
Yes, and then you just find something, and then you just are like, yes, I want this, and then you touch it, and then it's like pops to some other corner of the tray where you have the sample in it, and you have to start searching again.
Oh my goodness!
So this needs a lot of concentration. This work,
right?
But that is only as I said, I have 3 different types of samples I'm working with.
Mm-hmm.
So it's always forums, but from different sites or different sampling methods. So besides the nets, we also have so-called sediment traps, which is like they are anchored at different depths in the ocean, and they are there for the whole year, and they just have some cups where they are collecting what is sinking downwards. So it's not like the net where we put the net in a certain depth and take what is there the minute we are sampling, but there we really trap everything that is sinking downwards, falling into the cups.
Okay.
And by that we can get a better impression on what is happening throughout the year.
Oh, because it makes levels, right? It'll make like layers. Oh, sorry about that.
No problem. Yeah. So what we can so I'm working, for example, with sediment trap samples at a station where we have one trap that is at about 400 meter depth, and then one that is at 2,000 meter depth. So we can also see what is sinking really down. Mm-hmm. And then the advantage is that we can put 20 cups on this device, and after 15 days, or in summer and winter when there's not much going on, one can also put it on 30 days. It moves. So then the collection, it is collecting material in one cup for 15 days, and then the next cup is used. So we can really see, okay, in this 15 days, from the 1st to the 15th of August, there were so many Foraminifera, and then from the 15th August to the 1st August or something, there were so many, and really understand what is happening throughout the year.
I love it.
And that is really helpful, because in the Arctic, usually the net samples we only get from summer period, because in the winter it's a bit difficult to get into the Arctic with the ship.
Yeah, there's it freezes over in places.
Yes, and for that, those stations are very helpful, because there's also not much going on in the winter time, so there are really not many Foraminifera, because they are not producing new ones, and just stay until the conditions get better and they can find more food, and then they start producing younger ones again. But still, we can get a better impression from that.
I totally get how you get the samples now. The plankton nets, I've used those before, actually. Before the pandemic, I took the kids, or my students, a group of students, to the westernmost side of Vancouver Island, and we hopped on, we did all these cool things on the oceans, and one of the things we did was we went on a boat and took plankton samples at the mouth of where a river went into the ocean, and then we went out into the ocean and took plankton samples. And the kids thought that was amazing, because they got to haul the net up, and the plankton looked completely different because it was different depths. And that's exactly how you explained it. So that's amazing. I made a bad bet. I bet the kids that they couldn't find a plankton that looked like plankton. I'd give some kid $5 if they found a plankton. And some kid found it in 30 seconds, because I didn't know that those zooplanktons that looked like plankton were super common. So that was my mistake. Some kids found it, so I had to give this kid $5 for finding the plankton.
And they probably spent that $5 in that store that they love in Banfield.
Right. So this is Chris, my wife, and Chris, you took kids to Banfield too with your school.
Yeah, we sure did. I do have a question. You were talking about how they, through their chemical reaction, produce carbon dioxide or release carbon dioxide, but are they performing photosynthesis as well? Like, are they a phytoplankton?
Not so themselves, definitely not, but there are some that incorporate some algae cells and do some symbiosis. So use them as symbionts, and then the algae cells inside them do some photosynthesis. But I think it's not that common for the Arctic species. So it's depending a bit on what species you have. Okay. But of course, those then also do photosynthesis, and that of course then is another factor why it plays a role for the carbon cycle. Yes.
These guys eat stuff, right? These organisms eat things to stay alive. They're because they're a protist / amoeba thing, I think. Is that right, Fransika? Yes, I don't know.
yes, they can eat stuff actually, but the sad thing is that we are still not really sure what exactly they eat.
Oh wow!
Even because it's really like you cannot just do observation in the field.
You can't see them having a snack.
And it's a bit difficult, and in the lab, they are not that happy. It's really not that easy to culture them in labs. So at some point, people discovered in cultures that they are happy when you feed them with a certain type of something like a tiny shrimp or so, I guess, which was one that is from freshwater lakes. So which they knew for sure is not their real prey because they live in ocean water and not in freshwater lakes.
Wow.
But that worked. But so a colleague of mine is now doing, and some other colleagues also from Norway are doing some cultural experiments at the moment, and also try to figure out a bit like what do they eat, how does it work, and it seems a bit like they eat like everything they can find. But
they're not picky.
Not really picky. I mean, they cannot really move directly into some direction. They can just like somehow, they only have this one cell. They can which is a bit crazy. They can grow like tiny little, it looks a bit like spaghetti that is going out of their body, which is like extension of their one body cell.
Yeah.
And with that, they can just when then something is floating into that, they can bring it inside of their body.
Oh, so it's like a octopus tentacles bringing it towards a mouth, sort of, but not really.
Sort of, but a little bit more passive,
yes, right? But, wow. I just image searched these Foraminifera, these forums, and they do look like popcorn. They look like cross between a popcorn and a brain. They're wild looking things.
Yes.
So one more question before we open the floor to our audience to maybe pick your brain about some stuff. Are you okay with that, Fransika? Having some questions thrown at you? Okay. And again, it's really important for the Bunsen and Beaker stuff is, do you have a pet story you could share with us?
Yes. So I was thinking a bit like I had several pets in the past, and I actually also listened to the podcast we did before because I wanted to check what did I tell you there, so I didn't want to tell the same thing.
Okay.
But I decided I think we already also talked about it during the second last pet chat. So I have guinea pigs, and I have had guinea pigs all my life. So I started having guinea pigs in 2000. Which is now 20 years ago, which is crazy when I think about it. So I really had guinea pigs all my life. And I think there was a very short period of a couple of weeks when one died and I didn't got a new one directly. And then I got 2 new ones. So I actually have guinea pigs for 20 years now, 22 years now. And so there are, of course, a lot of stories I could tell, but maybe I will tell a bit about the the ones I have now. So I had an intense time with them together the last 2 years, because we are now sharing the office.
Oh, they're your office mates.
Because like I'm stuck in home office most of the time. Every time I'm not in the lab, I've been in home office mostly since the beginning of the pandemic.
Mm-hmm.
And so we moved in spring 2020 to another flat, and there now the area of my guinea pigs is in the same room as my office, so my desk. And so it's very nice, because you have some, like, when you need a break from work, I can just turn around and take a look what they are doing. And like in most cases, they are, like, let's say 50% they are sleeping, the other 50% they are eating. That's guinea pig life. But what they are really good at is the timing of the day, because they really learned that at about 3 o'clock in the afternoon, we usually have some fruits, like, I don't know, have some apples, have a banana, whatever, like a snack in the afternoon. And they are really used to that now. So because they know, like, if we have apples, they get the inner part of the apple that we don't eat. And so they can get really annoying when I don't know, there's some reason for some reason I cannot give it to them. I don't do it directly at 3 o'clock. And also when I get out of the room already an hour earlier, like at 2, I decide, like, I want to get a tea. So I go to the kitchen and I make myself a tea, and then I come 5 minutes later back in the room with the mug of tea in my hand, and they are standing at their it's not a cage, but their area. So they are like, I have some plexiglass there, so they stand at the plexiglass and like panically moving their feet and like looking at me and like, okay, where is our apple now? And
they're begging.
No. Yes, absolutely. And
they
seem very stressed at that moment.
They're
like,
stressed.
Why have you no feet? Where's our food? And panicking and running around. And then I tell them, it's not 3 o'clock. You have to calm down. You wait. And when I sit down then and start work, okay, calm down very quick. But it's like when I get up in the morning in between and go to get a drink from the kitchen, whatever, they are not like that. So they really know the timing in which it's close to the time where they could get some apple.
I love it.
So they are very much learning those timings of the day.
Well, you know what? That's very similar to Bunsen and Beaker, because when Chris is eating an apple, they know they might get a little piece of it, and they're there right now. And same thing with I'm eating an apple. So dogs and guinea pigs.
Bunsen also knows when it's 3:35, because he'll come in here and he'll say, "Nope, you're all done work for today," and like try and pull me away from the computer.
That's a great story, Fransika. What are your guinea pig names again? I forget. I'm sorry.
So the names of the ones that we already talked before are Lumi and Mora. And I just actually only like 2 weeks before I left to Canada, I got a third one, which is called Bruno. And I will get to know him better when I will be back in Germany.
Oh, okay.
So he's now together with the 2 girls, and they get to know each other. And I just brought them to a friend to take care of them while I'm not there.
Gotcha. Hey everybody, thanks for listening to our show week after week. If you want to know how to support the Science Pawdcast, here are a couple ways. It's always going to be free to download, so you'll never have to worry about paying for it. But, things do cost money running a podcast, and here are a couple ways you could help us out. One is join our Patreon page. It's amazing. It's growing. It's almost like an extended family. There's multiple tiers of support, and we have lots of fun perks for being part of our Patreon page. The other way you could support us is giving us an awesome review on Apple Podcasts or Google Podcasts, anywhere you can rate our podcast. Give us a great review. The third way you could support the show is checking out the BunsenBurnerBMD.com website. We have awesome merch there. We worked really hard finding quality merchandise that's comfortable with vibrant colors. And you'll find in limited quantities over the next couple months, maybe even less, the 2022 Bunsen and Beaker calendar. So 3 ways to support us: the Patreon page, 2, give us a great review, 3, head over to our merch stop and see if there's anything there you'd like. Thanks, everybody. If you've just joined us for Spaces Unleashed, we have an expert guest that talks to us and tells us about their area of knowledge in a little interview section. We're going to open the floor now to questions from the audience. Fransika, you probably could answer quite a bit about microorganisms in the ocean and more about what you do. So if you have a question for Fransika about that, go ahead. I see Kiki, you came up, and then CC is on deck. So go ahead, Kiki, if you have a question.
Yes. Hi, my name is Kiki. I'm a coach, a mentor, and an educator. Thank you for this wonderful space, and thank you for talking about Foraminifera. And I'm glad to find out that they're omnivores, and I was looking it up actually because I'm a geek, and they have been alive for over 500 million years, which is like crazy. I love that you were talking about how tiny they are, and that they have the calcium bodies, and they're full of carbon. And I was looking up the very definition of the word because I took Latin in school, and I knew fere because it means ferno. It's Greek, well, it's Latin, but we use the word ferno, which is to bring, to carry. But foramen, my Latin is like really dusty. So it means whole, and I looked that up because I was, and I knew it was Latin immediately when I saw it. And my question is this, do we find many of these organisms when we extract petroleum, since they're that very old? So I'm guessing they have contributed to the petroleum resources that we have all over the earth. And where would we find most of them? I know that you're looking in areas that are colder, and now with the greenhouse effect that we're getting a lot of warmer summers, and that even affects the organisms that would typically be dormant. Has that changed their composition? So it's really 2 questions. Thank you so much for the floor.
Yeah, so thank you for the questions that are really interesting ones. So actually about the petroleum, I've never thought about it, but as one can find the foraminifera, you can find them everywhere where there's open ocean. And as you said, they have been on the planet since millions of years. So, and they are like sedimented at the ocean floor. So whatever is formed there at the ocean floor is also somehow foraminifera in there, I would say. And for the question about how many you can find where and the species diversity. So as I said, there are not that many species in the Arctic. There are definitely regions like when we get closer to the equator, where it's warmer, where there are more other organisms around, there are more plants, everything. It's the same with the foraminifera. Of course, the species diversity is much higher there. And I guess also in terms of the amount of material, like in total, but that is not really clear because there are also not that much studies on that yet. But they are definitely a lot more species. And in terms of the effect of the climate change, that is definitely true. So a colleague of mine have studied a bit the changes of the species that we can find in the Arctic. And we definitely see that when it gets warmer, then there are also like the so-called subarctic species. We at the moment find more when we go to the subarctic regions and the overlap between the Arctic and then the Atlantic Ocean, for example. There are some species that are also present in some warmer summer months in the Arctic, but not all over the year. And the numbers of those species is definitely rising. And this, of course, is then another question that it's interesting to tackle, to see if we have different species, do they also contribute differently to the carbon cycle? Because, for example, the most common Arctic species is one that has a really thick calcite shell, and there are other species that are not forming such thick shells. And if the species composition changes, such as that there are more species of the subarctic type that has a thinner shell, then of course also the carbon cycle could be changed in that way. So definitely climate change could have an effect. On the other side of when it's getting warmer in the Arctic, also it might be better to live there. There might be more shells to be formed. So there are a lot of options. So the change could be in different directions, or in the end have a zero result in the end because one is rising, the other is sinking. That's hard to say, but that's something we are working on somehow.
Thank you for that answer. So I'm guessing that this can change the research moving forward if we have a little more of the species coming out, more Foraminifera coming out of the ice. Maybe there's going to be more research in these locations. Thank you so much for taking the time to answer. It has been so informative. Thank you so much.
You're welcome.
Kiki, you're so good at saying Foraminifera. It just like rolls off your tongue. Have you been practicing it? Do you do that in secret, or is that because you speak multiple languages?
No, I don't speak. I have studied multiple languages. This is very different. And I did take 3 years of Latin back in high school. So yeah.
Okay, one second. Wait for it, and then I want you to say Foraminifera. Ready?
Foraminifera?
Yeah, there you go. All right. I struggled with that. You definitely showed me up there, Kiki. Good job. Great questions and great answers, Fransika. Hopefully, I didn't annoy people with the sound effects. Okay, so over to Cece. Cece, hello. Welcome up to the stage. Hello.
Thanks so much for taking my questions, and thanks so much, Fransika, for a fascinating space. So my question is about something you mentioned towards the end of your talk about how the Foraminifera, that's probably not correct, don't do well in labs. And so I was curious as to if anybody studying them has figured out why they don't do well in labs. Is it because we don't know what they eat? And then on a related note, are there other ocean microorganisms that do well in labs? And is there a reason why they do better?
I guess so it's not really clear if you would know why they don't do that good in labs. We could, of course, easily or try to change that. I guess, of course, like the food source, and that is, of course, a problem. But I guess another problem is just that it's hard to mimic the living environment of something that is living in the upper water column with the movement of the water and things like that. So that is, of course, hard to mimic. But I think we are also, so the things are improving there. So it's getting better. And in terms of other organisms, so I know what we are also doing in our working group, or what other people are doing that work with the benthic Foraminifera, so the ones that live on the ocean floor. The culturing of those in the lab is really no problem. So that works very nicely. They can also do they reproduce there and grow, and you can see that they are like moving up the walls, so you can see that they are still alive, and that is working very well. So with the Planktick, the problem is really that until now it was not really successful to have some following up generations. So what we can do is to catch some in the floating water, and then it also happens that they release some young new forams, but somehow it doesn't happen that they reproduce further until this far. But I also have to say that I'm not really into that myself, so I only know what I know from colleagues that are doing it. So as I said, I have one colleague working together with other people from another institute in Norway, trying to improve the culturing experiment. And I think they are doing a lot to figure out how to improve it. But I think it's really, the tough point is probably really that the situation is hard to mimic with like the motion of the ocean and in that direction.
That was really interesting. Thank you so much.
That is a great question, CC. I love how you rhymed your answer too at the end, Fransika. The motion of the ocean. That was really well done. Good job. Over to Dan and then to Pink. Hey, Dan, you got a question for Fransika?
Yeah, I feel like I just have to practice saying it though. Foraminifera, right? Right?
Foraminifera.
Wow.
Yeah, I'm terrible at it too, Dan. Kiki's got us beat. Wow. Kiki's got us beat so bad on it. So that's amazing. CC did well too.
Try it. Yes. Okay, one more time. I need to hear it one more time.
Foraminifera.
Foraminifera.
Yeah, it's getting better.
Foraminifera. Okay, gotcha. So I was just taken aback by this whole description you had of the way they consume energy. You know, they're this like spaghetti-like tendrils. Now they're monocellular, right?
Yes.
That seems just a little magical to me. So what is that process like? How is that? Is that something that's like controlled by the nucleus and it just says, hey, there's something floating by, let's eat it? Or like, how does that work? I'm so mystified by that.
I have been sitting here wondering the same thing, Dan, and I've been embarrassed to ask. So thank you.
I am that guy that asks those questions.
Yeah. It's no problem. So it's really funny because I realized that before when I talk to people from outside the field about forums that they often ask about how they live and what they do. And I'm always like, I don't know, because actually I don't, like my research focus is not the living species, but the shell. So I really don't, like, I know some things because I read and I work in a group where other people work with other stuff, but I also never think about those questions, which makes it really interesting for me that I'm asked it, because then I have to think about it as well. So as I said, it's really amazing. They have this one cell and somehow they can like also move into different parts of their shell and then have this spaghetti-like things going out of the shell and into the water. And I guess what they do is not really that they realize that there is some food coming around, but it's more like they are more like, okay, I'm hungry, so I put them out and hope that something floats by and I can get it. But that is more, I would guess it works like that, and more, I know that it is like that.
That does not work for me on the couch. I'll tell you that. That does not work. Like I can't just flail my arms around and hopefully food comes to me on the couch. But go ahead, Dan. Sorry, I had to be snippy there.
That was brilliant. Well, I have something kind of snippy too, but I mean it just reminds me of a Venus flytrap. Like it just is sitting there and it's biomechanical and it's just kind of waiting for something to happen to come by, and so it's fascinating to me. I need to like do some research on that on my own. I don't know why I'm so fascinated by it, but thank you.
But you should definitely check out like in the internet some pictures. So like for example, I just checked when you go to Wikipedia, which is a totally non-scientific source, but for looking things up, it's really nice. So when you just type in foraminifera there in the English article, the first image you see is a really nice image where you can see this nice
oh my goodness,
how it lives.
It's so there
you can,
I see it.
You can see it. So that's really great. And I'm just thinking, I know I'm not sure because I have seen videos of those, like how they move, but I'm not sure if the videos are like, if the people from my group have the videos because they have taken them themselves, or if there are also some videos that are published like on YouTube or something, but I can take a look, and if I find them, put it later on Twitter. So yeah, but that's really amazing how it looks like. So, and that is always like when I think, as I said, I usually don't look at the living beings, but when I see pictures or videos of that, it's really amazing how simple they are and still how they look like and how it works.
I'm just tweeting that picture, and I'm going to throw it into the nest for people to look at. It's so alien. It looks like Pac-Man with some kind of like weird bad hair day. I don't even know. I don't even know, Fransika. Okay. Oh, I have Twitter Blue. It's just waiting for me to okay that I haven't made typos, which I make even though I have Twitter Blue. 1 second here. I'll throw it into the nest. There we go. So this is what you're talking about, right, Fransika? It's going to show up top there in the nest. That's a forum with the spaghetti tendrils. Did I get the right thing, or should I delete it?
Yeah, I don't see it yet. But
you don't. It might be
I'm
delayed
checking.
a bit. It'll be up in the nest. I put a tweet of yours and then the picture of that. Oh, I also have a sound effect for putting things in the nest. Okay, so if everybody looks up to the nest, you can see an alien forum up there. Do you see it now, Fransika?
Yes.
Look
I
at
see
it.
and it's absolutely right. So that is what you can see. So in the center, you have the shell, and then you see also this outside sparkling thing, which is a bit like, I guess, the reflection that you can see that the shell is really thick. Mm-hmm. So this white looks like a halo a bit. And then you have those spaghetti-like things that are like tiny parts, and they can just really, when there's something stuck in there, they can move it inside.
What? I'm going to stay awake tonight thinking about this. Probably it's because I had coffee too late. Okay. I want to respect I want to respect your time, Fransika. Do you have to go at the top of the hour, or do you have another
No, I have nothing to do here besides going to bed at some point.
Okay, all right.
But it's not has to be that early. But I just actually found that there's on YouTube a video how this works with the feeding. So I can answer. I put it in the answer to your tweet.
Oh, okay, sweet. All right. Oh, you've put it on Twitter? Okay, so YouTube. Orbulinia feeding on Terminia. Okay, I don't know.
Yes, so
okay, you know what that means. I don't.
Obulina is the name of a species.
So okay.
That is the species that is feeding, and Artemia, that is the thing you can see there. That is the living thing that is eaten. That is, as I said, this freshwater shrimp thing.
Okay, I've put that into the nest for everybody to take a look at. It's not coming up for me. Like I don't know. It could be because I'm on my phone, maybe, but it is. The YouTube link is up there.
Yeah, so I can see that in the nest.
Oh, okay, perfect. Good deal.
That works. And there you can really see how it moves.
Wow, that is wild. Somebody just said it looks like something. It looks like something in outer space. It's so wild. Okay, Pink, you've been very patient. Do you have a question? Go ahead. Go ahead, Pink.
Okay, I'm fascinated too, but I have like a different subject. Why don't they respect the fishing rights of Native Canadians?
Oh, I don't know if Fransika can answer that, Pink. That's a great question, but that's not what we're talking about tonight. And I do respect and honor what you said, but that's not something that we're talking about tonight. Thank you for your question, though.
Well, I'm fascinated, so can you tell me more, please?
Oh, about the organism? Yeah, just listen along, you bet. Fransika, is there something from your, like, just to kind of, if there, oh, and one more thing, if there's anybody else that has a question, go ahead and request the mic, and we'll get you up here. If not, that's fine. I have another question for you, Fransika. What is, it's a tough one because I know you're in your PhD studies right now. Is there an end date for your study? Like, are you hoping to get enough data to make some kind of conclusion? Are you hoping for when coronavirus kind of like dies out, you can go on a ship? What's your future plans with your research?
Well, a bit of all of this. So first of all, I really hope that I can this summer finally go on a ship. So after there has been, since I started now, I think 3 or 4 research vessels that I was like thinking about, or we were thinking that I could go on. Finally, there will be one from the Norwegian people I mentioned before that do a lot of this culturing thing. That will happen this year in summer, end of June, beginning of July, and that would start in north of Norway in Tromsø and then go further north in the direction of the Arctic for about 2 weeks. So that is what I'm really hoping for, and not in the matter of getting further data, because I really don't need more material because I have so much already, but more in terms of to see the region and to see how the samples are taken and that matter. In terms of an ending date, this is a bit...
I know, that's a tough question. I'm sorry.
No, it's totally fine. So like, there is the official ending date at the moment. That is the end of September this year, where my contract ends.
Okay.
But so that is what we are a bit hoping for. There has been Corona all the time, and there has been in the last year some extensions from other people, from other projects that has been funding by the German research funding organization, which is also funding my project, which is part of a bigger Arctic research group. So we are a group of 12 PhD students in Germany, plus several PhD students and master students all over Canada, which is also why I'm currently here, because this is a project that is closely connecting between Germany and Canada. And so there is the hope that we would get some extension, but still I would like to try to finish up, like, as soon as possible. So probably until the end of this year, also a bit depending on what I want to do after, which I don't know yet.
Right. Well, you're young. You've done so much with your life already. You've got tons of time to make decisions. Are you enjoying Canada? Would you stay in Canada given the chance, or do you miss your home country?
So I'm really enjoying it. It's like great. It's so awesome to like see this, like, let's say, real winter. So because,
the real winter.
Yeah, I mean, like in Bremen, like when it's a cold time, we have like a couple of days when it's below 0 degrees Celsius, and so below freezing point. We have, like, last year we had actually the rare situation that we had about a week where we had snow, but usually it's more like 5 degrees Celsius above 0 and rain or drizzling all the time and very gray. So at the moment, all the people that I'm talking to at home are very jealous because we have so much sun here, which is amazing. I'm really looking forward also to get some experiences in terms of meeting more people and get out, which of course has been a bit difficult until now, but things are opened up a bit here. So restaurants have opened this week here again, and I hope that will be fine. But of course, I'm actually also very bound to my hometown. So I would also be very happy, of course, to be back home after the 2 months.
Yeah, I got you. It's very brave of you to come to a new country. I couldn't imagine leaving Canada and going to Germany. So kudos to you.
Yeah, well, so I've like, let's say I've trained. So during my master, I've been for 2 months close to Paris in France, which was already tough, but which was the difference, of course, is not that big. And the distance is not that far. So the thing that was frightening me the most was really the travel, because I've never left Europe before, never been on a plane so long, and never been on a plane alone. But it all turned out fine, even with the pandemic situation. So, and I'm very happy that I'm here now.
Awesome. Chris, did you have something?
Yeah, I'm just wondering, how many languages do you speak? Like, I think you speak French when you're in, France, but then also in Quebec, where you're in Montreal right now. I noticed you tweeted today, donc, bonne nuit. So I was just wondering how many languages you speak.
Yeah, so it's apparently English. I speak French, as you said, so I've learned French in school. I started in grade 6 with French, so 1 year after I started with English, and also had some final exams in French as well, so did a lot in there. And then it's German, so it's the 3 languages. And that's also one thing that I'm really happy about, that I had the chance to go to Canada and the French-speaking side, because I can then make use again of the language that I know. Because English I'm using during my daily work anywhere, also in Germany, but French is nice to use the language skill as well.
You're a triple threat. 3 different languages. So Fransika, I think we're at the end of the chat. There doesn't look like there's any more questions, and there's none in DM. I'll give anybody on the floor one more chance here. Chris or Kiki, you still are speakers if you have a question to go, and if not, we'll wrap up.
No, I don't have any more questions. I'm just fascinated by this entire topic.
So cool.
Me as well. I have no questions. However, it has been so absolutely interesting listening to it. And I thought, Jason, by me giving you the etymology of the word, you would come up with some jokes. I was hoping for that. I think that's why I actually did it, to be honest. But I was hoping for that. So. I was waiting and waiting.
Oh, I'm sorry. I was listening to you talk about the origins. I love that kind of stuff.
I know, because I geek out. But I
Yeah.
also gave you a little bit of a need for a joke. But okay, no worries.
Awesome. Oh, so somebody has a question. Do they? Okay, one quick question, Fransika. That just came in. Do these freeze in the winter? Do these planktic organisms freeze solid?
No, depending on where you are. So in the Arctic, they don't freeze. They just move where there's still water. But actually in the Antarctic, so it's the same species but a different genotype, I think. And there they know that they really freeze into the Arctic, into the Antarctic, into the surface water, ice, sea ice. And they live in there in some brine channels, and then defreeze again when it gets warmer, and then start reproducing again. But they are not doing that in the Arctic for whatever reason. So there is really some difference between those regions, even though we always tend to say, okay, it's from the terms of climate, it's rather similar, but the organisms that live there are different in some way.
Wow. You're getting some love in the DM. People are DMing me that they're loving the space, Fransika. So you did such a good job. If you're not following Fransika, please give her a follow. You just have to click on the little picture of her face in the parka thing there, and then click follow. That'd be great for her following count. I'm just going to get this going here. There we go. So we're at the end of Spaces Unleashed tonight. Thank you to our guest, Fransika Tell, who chatted with us about the Foraminifera, the forums. I don't know if I got it right, but I'll get it right eventually. I'll get it right.
Still get the F at this wrong point.
There we go. Thank you for the speakers who came up to talk. It takes some courage to ask questions. This Saturday, we have our second space. It's Spaces Unleashed Pet Chat. It's really wholesome. Anybody can come and chat. And some exciting news. On Thursday, Season 4 of the Science Pawdcast drops its first episode. So that's the first episode of Season 4 of the Science Pawdcast. All right. Take care, everybody. Thank you so much for coming tonight. I so appreciate it. And especially to my Spaces team out there showing up. I appreciate that too. Fransika, one more time. Thank you so much. I have a million follow-up questions, but we better call her a night.
Yeah, thanks a lot for having me. I had a lot of fun and really enjoyed talking to you. And maybe for a goodbye, just if you want to learn more names, I can just give you the name of the species I'm working on, because in contrast to that, Foraminifera is easy to spell because it's called Neogloboquadrina pachyderma.
How many points is that in Scrabble?
I don't know.
I think you win. I think you just win.
A lot.
Yeah, a triple word score. Fransika,
So
you're
that is the Arctic species I'm working on. And yeah, I really enjoyed talking to you. And also, as I said, the questions, that is really interesting to have questions that one is not thinking about themselves, but it's really nice to have them.
yeah, you're an all-star. You're such an all-star. I can't wait to see what happens with your career. Take care, everybody.
Thanks a lot.
For science,
you.
you betcha. For science, empathy, and cuteness.